The bulk of the human genome contains many sequences that do not code for any proteins. These regions of the genome are sometimes referred to as junk DNA and make up more than 98% of human genome. Included in the non-coding DNA, there are many repetitive DNA sequences. Two important families of repetitive elements are the lines, long interspersed elements, and the signs, short interspersed elements. L1 is the most prevalent, line, and makes up about 17% of the human genome. Alu is the most prevalent sign and equals about 11% of the genome. Alu and other signs are defective transposons. They depend on the enzymes of other transposon, like L1, for mobility. It is estimated that there are about 75,000 L1 and 1,200,000 Alu elements distributed throughout the human genome. L1 is approximately 20 times the length of Alu, encodes all the molecular machinery needed to replicate and move within the genome. An L1 element is approximately 6,000 nucleotides in length, with 2 untranslated regions, UTRs, and 2 open reading frames, ORFs. The ORF protein products assemble into a complex that enables L1 and other elements such as Alu to move throughout the genome. The structure of L1 is a stark contrast with that of the tiny 300 nucleotide Alu. Alu elements have no open reading frames and so encode no proteins. Alu elements are characterized by a sequence with 2 GC-rich regions, the left and the right monomers. These monomers are connected by an A-rich linker. Alu elements end with a poly-A tail. In genome, Alu elements are immediately flanked by AT-rich sequence. The left Alu region contains 2 specific features, box A and box B, which are binding sites for transcription factors and RNA polymerase III. Alu elements are transcribed into RNA by these proteins until they reach a stretch of T's in the genomic DNA. The Alu RNA can act as template for the formation of a new Alu element that can insert itself in a new genome location. A likely model for this process requires the enzyme, a reverse transcriptase, encoded by L1. In addition to reverse transcription, L1 reverse transcriptase, RT, has the unique ability to nick DNA in a site-preferential manner. Most often the nick is made at the consensus sequence TTAAAA. This can create a single-stranded sequence of T's that hydrogen bonds with poly-A tail of the Alu RNA to form a short RNA DNA heteroduplex. The heteroduplex, then, can serve as a primer for L1 reverse transcriptase to synthesize a complementary DNA or cDNA strand. A second nick is made on the opposite strand, a variable distance from the initial cleavage site. Then, either the L1 reverse transcriptase or a cellular DNA polymerase synthesizes the second DNA strand. This results in an Alu element inserted into a novel position in the genome. The insertion also creates a direct repeat sequence on either side of the element. Alu elements are short interspersed elements, signs. These are about 300 base pairs long. They are named after the Alu restriction enzyme that recognizes a site within their sequences. These are retrotransposons, which means they copy themselves through an RNA intermediate and insert back into the genome. These are only found in primates. Humans have over 1 million Alu copies.